Hello, everybody.
Hello.
Happy Monday. Just let everybody.
My name is up there.
Oh, yeah, I can see it. All right, if you would, go ahead and type your first name or your nickname into the chat for me. Thank you. Still missing just a couple people, but it's just 4 o'clock, so. Oh, thank you, Debbie. Thanks for letting me know. I'm sorry to hear it.
Yeah, she'll try to get on, but otherwise she might have to access the recorded
Okay.
lecture.
Okay.
Also, I'm gonna have to leave at 5 today.
Okay. That's not a problem. Thanks for letting me
Okay.
know.
Hey, and I have another question. So was that just like was it like in there twice the midterm? Because did you see what I was talking about? Because after I went and looked again, it was taken, like it wasn't there anymore.
So what did you see?
So I saw that I had 2 midterms, like that I had, like one was part 1 and one was part, like I thought part 2 or something. And then when I went to click on the other one, it wouldn't let me access it, but it let me access that other one.
2 midterm, like midterm grades?
Yeah, like there was 2 midterm like lines that you could click on, and when it went to like one of them went to the test. So I don't know, I was like confused. I was freaking out. I was like, did I miss half the freaking midterm? And I didn't even know. But then you said that it's with my other class too. Like I'm having issues with things like that. Like there was a midterm thing in there, and she was like, she ended up emailing me and was like, oh, it was just a mistake. But like, I feel like this whole technology thing is like new to everybody.
Well, I'll tell you what we're learning is where the bugs are in the technology, right?
Yeah.
We're really learning where all the bugs are.
Yeah.
So yeah. But the other thing to remember is sometimes, and not that this is necessarily what happened, but you also have to remember that when a professor makes a change in the software, in the Canvas software, it takes a certain amount of time for it to update all the way through the system. So sometimes a professor will be working on something and will put something in to a module or something, and you won't see it right away. Or you'll see it, and then the professor removes it, and a few minutes later, it'll be gone. So you have to remember that it does take a while for what's happening on the professor's end to populate the entire system.
Mhm.
And so sometimes things appear and disappear, which is bizarre, but
Dr. P,
also,
I think I saw that too. It was due, it had the same title, but it was due in June because it told me it was missing and I panicked. I was like, oh my God, it didn't go through.
oh. So
But it did, and
that's
then it
the other
went
thing
away.
to remember is when you, for this course anyway, not necessarily all your courses, but for this course, the best way to access files is in modules. So go to the modules, scroll down to the week, click on the exam. It's
Yeah.
possible if you go to like quizzes, if you click on quizzes and you look for the particular exam, it's possible that there'll be an old copy
Yeah.
of an exam that doesn't exist anymore.
Yeah, I go to grades usually and I click on those.
Yeah.
But I don't.
But if you're trying to go into an exam, if you're trying to take an exam or you're trying to take a quiz, best to go in through modules because you'll know that that file is the one that was most recently put in by the professor, at least by me. If
Okay.
in some courses you can click on a link that says files, and you're going to find like old files that the professor has deleted from the course. So I always tell students, don't go in like back doors to find examinations and quizzes and things in Canvas, because there are too many places where old things can sort of linger in the system. So at least for me, when it comes time to take a quiz or take an exam or take a lab practical, the very best way to do it is to go click on the modules section, scroll down to this week, and click on that link for the lecture exam or the lab practical, because that's going to be your most current copy of the exam. Yeah, you know, as a professor, we learn stuff as we go too. And what I've learned teaching remotely is that it's going to be a good practice for me to just go through Canvas like once or twice a year and just try to get rid of old things that are still in there in places that can confuse students, that can still show up and confuse students. But yeah, we're all learning how buggy technology can be. We depend on it so much, but it's not perfect, not by any stretch of the imagination. I'll tell you what, though, I teach at another school that is using Blackboard. I don't know if you've ever used Blackboard, but it's like Canvas. It's an organizational tool for a class that's being taught remotely. And Canvas is just leaps and bounds better than Blackboard is. Blackboard
I
is,
used to use Blackboard. My internet's starting to break up too, just so you are aware.
oh, okay. Yeah, it sounds like it's not a good internet day for people today. All right. Yeah. So Debbie, that would have been an old version of the lab practical that was administered in the summer. And even though I deleted it, it may still exist in Canvas.
Out there somewhere. Yeah, that's probably what it is.
Very strange. Very strange. Yeah. All right. So today we're going to be talking about carbohydrates, and we're going to talk about carbohydrate catabolism, which is breaking apart carbohydrates in order to build ATP for energy. We're actually going to talk about carbohydrate catabolism this week in labs. So today we'll talk about a couple of tests where we look at different pathways that can be used to break down carbohydrates. And then the next time we get together on Wednesday, we're going to talk about fermentation specifically. But before I get into the lab material, I did want to just mention to you that your pathogen assignment was returned to you a couple of days ago. Hopefully you've had a chance to see that. If you haven't, go into Canvas and click on the rough draft submission link so you can find it and see my comments. Basically what I do is I download your first draft, and then I make my comments directly on your first draft. So it's a little bit different from quizzes and exams in that I'm going to actually be typing onto your first draft, and then I send it back to you. So you'll find it attached to a comment, basically, that I made about your submission. And I sent out an announcement because it's important that you know, as you're looking over your first draft, that if I ask you to do a little more research about something, I don't want you to feel like, oh, I forgot to put this into my first draft. That's not usually the case. I think all of you answered all of the questions that you were supposed to answer for your question group. But as part of this exercise, what I do, in addition to asking you to revise some, structural things in your answers, things like fixing sentences or choosing different wording or somehow altering the structure of your answers. The other thing I do is I send you off trying to find some more information. So I may pose a new question to you and ask you to do a little research and see what you can find out. That's just part of this assignment. This assignment is designed because it has the two submissions. It's designed not only to have you do this work to learn about this particular pathogen, but also to get practice responding to editorial criticism. Learning how to revise your work according to someone's comments. It's a good chance to practice that, because when we go out into the professional world, depending upon your profession, You often have to write reports. You have to write summary reports, or you have to keep patient charts, or you have to do all kinds of written things in your job. And from time to time, your supervisor, whoever that person is going to ask you to hand them written material. And that written material may be returned to you with comments asking you to change things. And it's a skill to learn how to revise, and to use the comments and that the person's given you and make revisions according to those comments. It's actually a real skill. And unfortunately, most people, most of us, don't get enough practice doing it. And so our writing skills can always use a little buffing, a little sharpening. And that goes for everyone. Everyone can always use practice with their writing skills. So I didn't want you to feel like if I asked you, for example, to tell me something else about medications that were used to treat tuberculosis, if that was the question you answered. It's not that, oh, that should have been in your first draft and it wasn't. It's that this is something else I want you to go look at. And I want you to respond, thoughtfully to whatever I've asked you to add in or perhaps change in your first draft. That's a part of this assignment, is how you respond and how you address these revision comments. The other thing, and I'll just say this just, from a practical standpoint, is in your professional life, it's really, really useful to learn how to take criticism, both in your work in general, but also in your written work. It's human nature when you write something and you hand it to your boss and you work hard on it. It's human nature to feel a little bit attacked when your boss looks at you and says, I don't like this, I want you to change it. And it's a really good exercise for all of us, again, to practice removing ourselves from the work and just remembering that that's all it is. It's just an assignment, it's work, and not to take criticism personally. I can say from my own experience, when I was a young veterinarian and my boss would review my charts and make comments about my charts, I remember feeling hurt because I thought I had done a good job, but he was just trying to teach me how to do a better job. And you have to learn how to not take things like that personally and view them just as professional growth. It's something that comes with practice. So anyway, I just wanted to make sure you didn't misinterpret me asking you to do a couple of additional things in your final draft. And again, if anyone has any questions about any of the comments that I made, if something doesn't make sense to you or you're not quite sure what I'm asking, feel free to get in touch with me and we'll message me and we'll figure it out together. So that final draft is due a week from Wednesday. So you have until March 31st at midnight to get the final draft submitted to me. Okay. All right. Oh, and overall, the first drafts were very good. Everyone did very well on the first draft, so I was very pleased with them. Good job. All right. So what I'm going to do now is go ahead and jump into the lab material for today. And I'll start off by saying that when we talk about carbohydrate metabolism, and remember that word just means, when we talk about metabolism, we're just talking about chemical reactions that are used by cells to break things down or build up new molecules. And of course, when we break down molecules, we call that catabolism. Those are catabolic. And when a cell builds up new molecules, that's called anabolism. Those are anabolic. So we kind of break metabolic reactions into two groups, whether something's being built up or something's being broken down. So when we talk about carbohydrate catabolism, we're talking about how microbial cells, all cells really, break down glucose, and they use it to build this really important molecule called ATP. It can be a little hard when we talk about carbohydrates and carbohydrate metabolism, because the term carbohydrate is really kind of weighted in our everyday language. We use that word all the time, just in ordinary language. So it's not limited to discussions of science. And unfortunately, in our ordinary language, the word carbohydrate has gotten kind of a bad reputation because of the kind of things that happen in diet culture. A lot of folks hear the word carbohydrate and they think, oh, carbohydrates are bad for us. But of course, carbohydrates are just one of the macromolecules in our cells. And they are absolutely essential to living things. And I'm sorry. That's all right.
You're saying catabolism and analysm, and you keep switching whether they build up or break down.
So
Catabolism is breakdown.
That's right. And anabolism with an A
right
is building up. So anabolic would be a process that builds a molecule.
All right, you just were throwing those words out there at the same time, and I screwed up.
So, so people, so what I wanted, my point was that glucose happens to be sort of this universal carbohydrate that all cells are able to use for this process that we call energy. And remember, when we use that term energy in science, we're talking about the ability to do work. When we say that a cell is going to use energy, we're talking about a cell making use of a molecule called ATP. And it's using that molecule because that molecule has potential energy stored in it, in its bonds. So when we talk about microbes breaking down glucose in order to build ATP, another way of saying that is that they are using the carbohydrates for energy. And again, this is terminology or speech that people use all the time in everyday language. They'll say, oh, well, that carbohydrate has a lot of energy in it, or it gives you a lot of energy. But we have to remember that what we're really talking about is building ATP, because cells use ATP as energy currency. So all cells can use glucose for energy currency in one way or another, but they use it in different ways. And so we can examine those ways, those pathways, in order to learn characteristics of microbes. And if we have a microbe we're trying to identify, we can test its ability to catabolize or break down carbohydrate to give us an idea of what organism it is, just like all the other features we've been talking about in organisms. So I'm going to go ahead now and open up our slides for the day. And that's what you should be seeing in front of you right now. A slide that says Lab Carbohydrate Catabolism. Now, our objectives today first are to talk through some vocabulary, because again, a lot of this terminology is used in everyday language, and unfortunately, it's used incorrectly. So we need to put some definitions on these terms that we're going to be using. Then we're going to talk about the difference between what we're going to call oxidative catabolism and fermentative catabolism regarding carbohydrate. And then finally, we're going to do an experiment. We're going to perform two tests that are used to evaluate carbohydrate catabolism. One is called the Starch hydrolysis test, and the other one is called the OF test. And the OF test it stands for oxidative-fermentative test. So let's talk through some of this vocabulary. This term carbohydrate, if you break this term down, if you translate this term, it means carbon, water. So when you think of the 4 elements that we've discussed that all living things are primarily made from, remember that's carbon, hydrogen, oxygen, and nitrogen. 96% of all living material on our planet is made out of those 4 elements. When we look at carbohydrate, one of the 4 macromolecules, it only has 3 of those 4 elements in it. It has carbon, hydrogen, and oxygen in it. And when you look at how carbohydrates are structured, when you look at their molecular structure, what you essentially find are combinations of carbons and water molecules bound together to build this thing that we call a carbohydrate. So it's essentially carbon and water that come together and build this molecule. Now, some of the confusion comes in because we can also refer to a carbohydrate as a sugar. Those 2 terms are interchangeable in science. The problem comes in when people hear the word sugar, because we often associate that word with table sugar. So most people, when you say the word sugar to them, they think about white granulated table sugar. And of course, table sugar is only one of many, many types of carbohydrates. So it's kind of unfortunate that we use this term to mean a big group of carbohydrates when the general public doesn't understand that distinction. Now, there are many different carbohydrates and we can break them down into groups based on their structure. So when you hear the word monosaccharide, this translates as one, that's the mono. Sugar, that's what saccharide translates into. One sugar. In other words, a monosaccharide is one molecule of a sugar, a carbohydrate. So something like glucose is a monosaccharide because that structure, that carbohydrate structure is just one individual molecule. We can also refer to a monosaccharide as a simple sugar. You'll hear that terminology too. And just if you're not familiar, this word dextrose, which we see a lot in medicine, dextrose is just glucose. It's essentially the identical molecule. Another simple monosaccharide sugar is fructose. You've probably heard of fructose, that's fruit sugar. Now, a disaccharide, the di part means 2. So this is 2 sugars that are bound together, that are connected to each other with a covalent bond. And a good example of a disaccharide is lactose. And lactose is the carbohydrate, the sugar that's found in milk. All lactose is a molecule of glucose and a molecule of galactose that are bound together. So we put a covalent bond between them and we get lactose, a disaccharide. Sucrose, table sugar, is another one. Table sugar or sucrose is made out of glucose, one glucose, bound to one fructose. Together, they create sucrose. And maltose is another one you'll see very often on food labels. Maltose is just 2 individual molecules of glucose bound together. So you can find disaccharides and monosaccharides in all kinds of microbial cells because these are the kinds of carbohydrates that are very small and very usable. They're easy to break down because they're more simple molecules. Now, that simple word is another place where the general public can sometimes misinterpret terminology. When we in science talk about a simple sugar, we're talking about either a monosaccharide or a disaccharide. So any of these examples that I've given up here would be considered simple sugars or simple carbohydrates. Now, the opposite of a simple carbohydrate is a complex carbohydrate. And we'll get to that one in just a minute. Now, after monosaccharides and disaccharides, the next most common term you'll hear is oligosaccharide. This term oligo translates as few. So this means few sugars. And by few, we're talking about greater than 2. I should change this. Greater than 2, but less than 20. So anywhere between 3 and 20 individual monosaccharides that are bound together will create what we call an oligosaccharide. Now, we don't talk about oligosaccharides in our common language. We don't talk about that in our everyday language because oligosaccharides are not really dietarily relevant. Where you tend to see these are in cell structure because cells like to put oligosaccharides on the surface off of the membrane in order to help identify what kind of an organism it is. So these are the kinds of carbohydrates that are used by cells to identify themselves. Finally, we have a polysaccharide. Poly just means many. And we're going to define that as a carbohydrate that has more than 20 sugars in it. A good example of a polysaccharide is this molecule called starch. All starch is a whole lot of glucose molecules, all bound together to create this polysaccharide. The plant makes the starch because it's a good way to store glucose. It's a good way for the plant to store glucose, so it has it available when it needs it. You and I also store glucose as a polysaccharide. We don't make starch, we make a molecule called glycogen. So your muscles, for example, are very good at producing glycogen. That's just many glucose molecules, greater than 20, all joined together to create this big polysaccharide that we call glycogen. Polysaccharides are also known as complex carbohydrates. So when you hear this term, complex carbohydrate, we're talking about polysaccharides. There are many other polysaccharides in nature. Cellulose is a polysaccharide. Chitin, remember we talked about chitin. Chitin is a polysaccharide. Peptidoglycan is a polysaccharide. So we see them all over the place in nature. What we're going to be focused on today, though, are simple sugars, and primarily this simple sugar called glucose, because this is the monosaccharide that all cells can use in one way or another in order to make this critically important molecule called ATP. Now, when we talk about the catabolism of carbohydrates, one of the tests we have available to us is this test that's called Starch hydrolysis test. Microbial cells contain what are called hydrolytic enzymes that can break apart carbohydrates in the environment in order to get at the glucose. Now, all this hydrolytic term means, if you translate this term, hydro means water, and lytic means break down or break apart. So these are enzymes that use water to break apart carbohydrate molecules. And again, if they're breaking them apart in the environment, what they're trying to do is take a larger carbohydrate and break it down so they can get monosaccharides or disaccharides out of it. Hydrolytic enzymes inside microbial cells tend to be one of two types. They belong to one of two families. Some of them are called exoenzymes, and some are called endoenzymes. Exo meaning outside, and endo meaning within. So an exoenzyme is a hydrolytic enzyme that the microbe is going to secrete out into the environment. And a really good example of an exoenzyme is amylase. Microbial cells, some of them, are able to secrete amylase out into the world if they find themselves in an environment that has a lot of starch in it. They can secrete amylase to break that starch down. And remember, starch is made out of many, many glucose molecules. So if they can break the starch down in the environment, they can then absorb the glucose and use it to make ATP. You and I also have amylase. We also use amylase to digest starch. It's just that our cells secrete it into the internal regions of our body. We don't secrete it out into the environment like microbes do. Endoenzymes are different. Endoenzymes are the kinds of hydrolytic enzymes that a microbial cell keeps inside of itself in order to break down a molecule. So those are different. We're going to focus on exoenzymes, specifically amylase, with this starch hydrolysis test. And this is what the test looks like. It's an agar plate that contains a nutrient medium, just a basal medium, that has had starch added to it. So this is just some very basic growth medium, like a basal medium, that we've added starch to. And as you can see, it's very boring looking. It doesn't have any interesting colors or anything. It's just this sort of beigeish color. So what we would do with this starch plate is we would take our cells, whatever kind of microbial cells we are interested in studying, and we would spread them over the surface of this agar, just like we always do, and put it into the incubator in order to grow the cells. And then the next day, when we take it out, we're going to do something very important to it. We're going to add iodine to the plate. We're going to pour iodine on the surface of the plate. And when you do that, what happens is the iodine binds to any starch that is still on that plate. And it turns that starch a black color. And that's what you're looking at here. So this is a starch hydrolysis plate that there's no cells growing on. So this one has not been inoculated. They just took the plate and they put iodine on it. And as you can see, the whole plate turns black. Anywhere that there is starch in the agar, the iodine is going to turn the starch black. Now take a look at the picture down here at the bottom. Here we have two starch hydrolysis test plates that have had iodine added, showing us a positive and a negative result. As you can see, there aren't colonies growing on these plates or anything like that. We just have this big, thick streak down the middle of the plate. So somebody took an inoculating loop, dipped it into the broth culture, and just put one swipe down the center of the plate. And they did the same thing over here. Now, remember, the plate originally looked like this one. It was originally just this, very basic looking agar plate. they would have streaked the cells down the center, put the plate in the incubator, and left it overnight so the cells can grow. That's what you can see right here. You can see this sort of yellowish, maybe cream colored line right here. Those are cells growing. Same thing over here. When you take the plate out the next day, you add the iodine. And the iodine is going to bind wherever there's starch and turn it black. So take a look at this plate. This is a positive result for starch hydrolysis. Here's the organism growing. Again, the cells are just growing in a straight line. You can see that there's black over here and there's black over here, but right around the line of cells, it's turned clear. In fact, it's so clear that when you put a piece of paper behind it, you can read the words on the paper. The agar is no longer sort of cloudy like it was here. Now it's clear. And the reason it's clear is because the starch is gone in this area. This organism, whatever it happens to be, secreted this exoenzyme called amylase. It secreted it outside of itself into the agar. And the amylase broke down the starch, at least in the area right around the cells. So if I were to test this clear area, I would not find any starch, but I would find glucose. There'd be lots of glucose in here because that's what starch breaks down into. I'd have to go out to this area over here to find starch. That's why it's still black. The amylase can only travel so far away from the cells. So they're only going to be able to break the starch down right in the area close to them. And that's why you get this clear streak. Now, if you look at this plate next to it, you can see a negative result. Here's the streak that the cells were, where the cells were placed. And you can see they've grown. You have this nice opaque sort of cream colored line right here. We took the plate out of the incubator and we added the iodine. And as you can see, the whole rest of the plate is black. There's no clear zone around this streak line. In other words, these cells did not secrete amylase because they can't. They don't have that ability. So this organism cannot break down starch. It cannot hydrolyze starch because it doesn't have the amylase enzyme to do it. So all of the agar around it remains black. It's full of starch.
So what is this test used for?
So this is just another way, another test that we can use to compare an organism to known information about it. If I get an organism into the lab, and I'll show you some examples in a minute. If I get an organism into the lab and I don't know what it is, but maybe I know where it was collected, or I know what the patient's symptoms were. This is yet another test that I can run. I can take that sample wherever it came from. I can spread it on that plate. I can add the iodine the next day, and I can look for the result. And now I have a piece of information about that organism. I know either it can or it can't break down starch. And again, in a textbook or other reference tables, I can use it to figure out which organism I have. Just like I could use the MSA plate, for example, to identify Staph aureus, or I could use the EMB plate to identify E. coli. I can use starch hydrolysis as well in order to identify certain organisms. Now, you couldn't just use the starch hydrolysis test. You'd have to use several tests. But it's one of the tests that we have available to help us identify certain organisms based on their ability to do this thing, to break down starch in their environment, because they can't all do it. Only some organisms can do that. Now, in addition to this starch hydrolysis test, there's another test that we're going to use in our experiment for this exercise, and that's this OF test. Remember, OF stands for oxidative versus fermentative. So this is an oxidative fermentative test. And what this test is going to do is tell us whether or not a microbe is capable of using oxidative catabolism to break down glucose or fermentative catabolism. I wrote metabolism here, but that's okay. To break down glucose, or maybe both. Maybe it's an organism that can do both things. And we're going to use glucose typically in an OF test, but I did want to mention that you don't have to use glucose in OF. You can use another sugar. It's just that most of the time when we run an OF test, we're going to use glucose. Because as I said a minute ago, we know all microbes use glucose in one form or another to make ATP. Now, a couple of important things to remember. When I use this term oxidative, this word does not mean oxygen. So oxidative is not the same thing as saying oxygen-based or involving oxygen. Oxidative refers to redox reactions, oxidation reduction reactions. These are a particular kind of chemical reaction. Oxidation reduction reactions are very common in living things. And so when we're looking at the ability of a microbe to use oxidative pathways, we're talking about oxidation reduction. Now, it turns out when we're discussing the use of glucose to make ATP, there are two oxidative pathways that we're really talking about, right? We're talking about aerobic cellular respiration or anaerobic cellular respiration. Both
Dr. P, could I just ask one question about this?
Yep.
Now, when we're talking about, it continues to say oxidative metabolism. Is that the way we should talk about it, or should we be saying catabolism?
Oh, okay. Remember, the term catabolism is just one kind of metabolism. So what that word metabolism with an M, all that word means is chemical reaction. So when we talk about metabolic pathways, we're just talking about breaking things down or building things up. We can distinguish metabolic reactions into two very big groups. One of them is the kind of reactions that build things. That's anabolic. The other one are the kinds of reactions that break things, and those are catabolic. So catabolic is one kind of metabolic. So on that slide, I should not have written metabolic because it's confusing. I should have just written catabolic. Yeah, because this is all about breaking glucose to build ATP. Some organisms break glucose using cellular respiration. There are two ways to do that, right? You can do it aerobically with oxygen, or you can do it anaerobically with some other molecule. Both of those, aerobic cellular respiration and anaerobic cellular respiration, are oxidative catabolism. Because both of those ways of doing cellular respiration involve oxidation reduction reactions.
Okay, thank you.
The terminology is very heavy in this. It's very heavy when we get into metabolism. There's a lot of words that kind of overlap with each other. So, when we're talking about the OF test, the only reason I even use the term oxidative is because it's in the name of the test. And we have to understand what it means. What do we mean when we talk about oxidative versus fermentative? Well, oxidative just means oxidation reduction. And there are two kinds of respiration that use oxidation reduction reactions. All right, let's jump back in here. We have to remember that there are actually two kinds of oxidative metabolism, oxidative metabolic reactions that can make ATP. There's an aerobic one, and there's an anaerobic one. OF testing only tests for aerobic cellular respiration. So make a note of that. Even though we call it oxidative versus fermentative, we're only looking at one of the oxidative pathways. And it's the one that's called aerobic cellular respiration. The other thing we have to remember is this. There are organisms that can do both of these, right? There are lots of organisms that can do both of these. There are organisms that can do aerobic respiration and fermentation. There are organisms that can do anaerobic respiration and fermentation. There are some that can do all three. Remember when we talked about facultative anaerobes? We said facultative anaerobes, they prefer to grow in oxygen. They prefer to do aerobic cellular respiration, but they can do anaerobic and they can ferment. They are very adaptive. They're able to do whatever makes sense at the moment in whatever environment they're in. So we have to remember that even though we talk about this as an either or test, there are some organisms that can do both things. All right. The other thing we should know, or you should make note of, is when I use the terms primary and secondary to talk about metabolic pathways, an organism's primary pathway is the one that it prefers, the one that it's going to do if it can do it. So, for example, you and I are aerobic cellular respiration organisms. As long as there's oxygen around, that is how we're going to break down glucose to make our ATP. If we don't have any oxygen around, we can't do cellular respiration. We can't, because oxygen is the final electron acceptor in cellular respiration. If there's no oxygen around, that process is going to stop. Okay. If we think about an anaerobic organism, in one of our exercises, we looked at an organism called Clostridium sporogenes, I believe. Clostridium is an obligate anaerobe. Remember, that's an organism that finds oxygen toxic. So an organism like Clostridium, it prefers to do anaerobic cellular respiration as long as it has its final electron acceptor available. And those organisms use things like nitrate or sulfate or carbonate as their final electron acceptors. So those are the kind of pathways that are primary metabolic pathways, sometimes respiration pathways. But those organisms can also, if they don't have their final electron acceptor, they can switch and they can do what we would call a secondary pathway, less preferred. And that would be fermentation. You and I, we can ferment, right? If you haven't gotten to this part of the lecture material, that's okay. But you and I are capable of fermentation. but it's a secondary pathway for us. We would much rather do aerobic cellular respiration. But if I'm a weekend warrior and I go out for a 10K run on a Saturday afternoon and I'm not in shape, what's going to happen? I'm going to start gasping for breath, right, as I run. I'm not going to be able to get enough oxygen to my cells to make enough ATP to do that work. My cells are not going to be able to do aerobic cellular respiration if I'm pushing myself to that level. So they're going to start fermenting and they're going to create lactic acid, and I'm going to feel terrible. But we're capable of that, just like many microbes are capable of that, capable of switching from primary pathways to secondary pathways. And of course, there are other microbes that don't have primary pathways. They can do whatever they want to do, whenever they want to do it. There are even some microbes that are pure fermenters. They only ferment. They don't even bother with respiration. They just ferment. So microbes are much more diverse metabolically than we are. And that's why it's helpful for us to run these kinds of tests, because there are reference tables available to us that tell us what each microbe that has been studied, what each microbe can do metabolically. So we can run these tests in order to figure out the identity of unknown microbes. Now, you've heard me say this before. I'll say it again, I'm sure, before the semester is over. Anaerobic cellular respiration is not the same thing as fermentation. Anaerobic cellular respiration is a long series of redox reactions. It's very similar to aerobic cellular respiration. It just uses a different final electron acceptor. So it breaks down glucose and it uses all of the electrons from glucose to make ATP. It's not the same thing as fermentation. Now, remember, we've talked about all the different categories we can put microbes in regarding their oxygen requirements. And these terms are very helpful to us for describing microbes in terms of their relationship with oxygen. But we have to remember that these terms don't tell us anything about whether or not a microbe can ferment. So we need to do additional tests to investigate whether or not it can ferment and what it can ferment. What kind of sugars can it ferment? Fermentation doesn't require oxygen, okay? But it can occur even when oxygen is present. Because just like in my example, sometimes an organism can't get enough oxygen in a particular situation to do aerobic cellular respiration. And if that happens, if it can't get enough oxygen in that moment, it will ferment. So fermentation can happen in the presence of oxygen. It just doesn't involve the oxygen. Fermentation can also happen when there's no oxygen around. All right, so you have to be careful not to get in the mindset that fermentation is no oxygen. Sometimes fermentation goes on because the organism just can't get enough of the oxygen it needs in order to do respiration. All right. So let's switch gears here and think about what OF medium really is. It's actually a basal medium. So remember what that means. We said basal or chemically defined. Those are the same thing. It's got a list of known ingredients in it. It supports the growth of a whole lot of non-fastidious bacteria. What we do is we take that basal medium and we add a lot of glucose to it, a large amount. In other words, more than the organism is going to need. Most of the time, when we use OF, we're using glucose. But like I said earlier, it is possible to modify the OF test in order to investigate other small carbohydrates, monosaccharides or disaccharides. Let's say I have an organism and I think it might be able to make use of, I don't know, sucrose, and I want to investigate that. I can put sucrose in my OF media instead of glucose. But most of the time, people do this test with glucose, and that's what we're going to use in our experiment. So a basal medium with lots of glucose and a little bit of peptone. We're going to put a little peptone in this media. Now, remember what peptone is. It's just partially digested protein. So it's a protein that we have already started to break apart in the laboratory, and then we put it into the medium. Now, why are we adding peptone when we're talking about carbohydrate catabolism? Well, the answer is the peptone is in there to support the growth of any microbes that cannot make use of the glucose that we're giving them under these conditions. Remember, we said we are testing two different conditions with this test. We are testing the conditions that would allow for aerobic cellular respiration and the conditions that would allow for fermentation. So if you're an organism that can't do either of those, you're going to be in trouble. You're not going to be able to grow in this medium because you can't make ATP. So what we do is we give them a little bit of peptone. That's going to keep that organism or these kinds of organisms alive in the tube, even though they can't use the glucose that we're trying to feed them. Now, OF medium also contains an indicator dye. This is one we haven't seen yet. It's called bromethymol blue. And this indicator dye is going to turn yellow in acidic conditions and blue in alkaline conditions. So OF medium starts off green, and I'll show you a picture in a minute. It starts off green. If the microbe that grows in it is capable of doing a metabolic reaction that produces an acid, it's going to turn yellow. And it turns out that that's the situation that occurs when carbohydrates get used to build ATP. If the organism cannot use the glucose in the tube and it starts using the peptone instead, that tube is going to turn blue. Now, OF medium is typically used as a broth, but you can also use it as a semi-solid material. Remember, we said one of the benefits of a semi-solid, adding a little bit of agar into the broth is because it can limit how much oxygen can get down into the broth. So we're going to use a semi-solid form of it today in our experiment. OF tests are always run in pairs. So you're always going to have two tubes when you run an OF test. In one of those tubes, you're going to have the OF medium open to the air. So you're going to put the cells in it. You're going to have that medium inside the tube exposed to the air. In the other tube, what you're going to do is put a little bit of mineral oil on top of the medium. Mineral oil is less dense than the culture medium, so it will sit right on the top. And what that does is it blocks any more oxygen from getting in. The open air tube is the tube that's going to test for aerobic oxidative catabolism of the glucose. In other words, cellular respiration. The tube that has the mineral oil on it is going to test for fermentation. So it's an OF test. We have to test for both things. And this is what this medium looks like. On this slide, you can see groups of tubes, always in pairs. We always run 2 tubes when we do an OF test. Over here, this is what the medium starts off looking like. This is that green color that the medium begins with. And you can see right here, this is just the medium in a test tube, and you can't see the open end, but this tube is open. It's exposed to the air. This tube has this on top of it. This is just a layer of mineral oil that we sort of drip down into the tube, and we layer it on top of the culture medium. And again, what that mineral oil is going to do is essentially prevent any more oxygen from getting in here. This is not an anaerobic environment. There's going to be some oxygen in this tube. There's just not going to be as much oxygen as there is in this tube. Now, if the organism can do oxidative catabolism of glucose, in other words, if it can do aerobic cellular respiration, it's going to produce an acid byproduct, and it's going to turn the tube yellow. And it's a very obvious yellow color change. If the organism is capable of fermenting glucose, we're going to see the yellow color under the oil tube. So in this pair of tubes right here on the left, the open air tube is yellow and the mineral oil tube is yellow. So this is an organism that can do both. It can do oxidative catabolism of glucose, and it can do fermentative catabolism of glucose. This organism in the middle, this is one that can do oxidative catabolism, but it cannot do fermentative. The media stayed green. So no acid has been produced in this tube. Had it fermented the glucose, it would have turned yellow. And over here, this is where we start with both tubes green. But if you inoculate the tubes and incubate them overnight, and they're both still green, that means the organism couldn't use the glucose. Now, I told you that there's peptone in this medium, and some organisms will use that peptone in order to stay alive. What you'll see is that the green turns blue. And this, let me scroll down here. This is the blue color I'm referring to right here. Not all organisms are going to be able to use that peptone, but we put it in there because some can, and it's going to be another piece of data about that organism. All right. Now, one of the questions we should ask ourselves about the OF test, since we're looking for the yellow color change, since we're looking for the production of an acid, is where does that acid come from? Fermentation, if you haven't gotten to this in the lecture yet, fermentation actually is a whole group of reactions. There are different fermentation pathways, in other words. Fermentation, though, always produces one or more products, including an acid. So sometimes you get an acid and an alcohol produced. Sometimes you get an acid and a gas produced through fermentation, but you always get an acid produced. And the acid is what's going to cause it to turn yellow. It depends on the organism, and it also depends on which sugar the organism is fermenting. Remember, on our MSA plate that we looked at, Staph aureus is capable of fermenting lots of sugars, including the mannitol sugar that was in that plate. The thing about fermentation, though, is you're always going to get some kind of acid produced. And as long as it's an acid, it will turn the media yellow. Now, what about the oxidative tube? What about the tube that's open to the air? What about the tube where cellular respiration is occurring? What acid is made during cellular respiration? Well, remember what cellular respiration is. It's a series of reactions that takes a glucose molecule and breaks it apart. It breaks all of the bonds in the glucose molecule. So it starts off with this process called glycolysis. The glucose is essentially going to be split in half to create a molecule called pyruvate. Then the pyruvate is going to go through a reaction called pyruvate processing, and it's going to be converted into what's called acetyl-CoA and a little bit of carbon dioxide. There's some gas being produced. And then the acetyl-CoA is going to go into what's called the Krebs cycle, and it's going to be converted to carbon dioxide. So during aerobic cellular respiration, in fact, during both kinds of cellular respiration, the glucose is being broken down into carbon dioxide. Now, carbon dioxide is a gas. And what happens in the cell is it gets reacted with water, and it becomes what's called carbonic acid. So this is where the acid comes from in cellular respiration. It comes from that carbon dioxide that gets made from the glucose. All right, a lot of information so far. So the key thing now is, do you understand how to interpret an OF test? Remember, there are going to be 2 tubes every time you run the test. You're going to put cells into both tubes. You're going to take your unknown, whatever it is, and you're going to put inoculate both tubes with cells. You're going to put the mineral oil over one, and you're going to leave the other one open to the air. And those go in the incubator. And then the next day, you take them out and you look at the color. If you see the yellow color, you know that that organism has created this acid byproduct with glucose. If it's the open air tube that turned yellow, that's an oxidative reaction. That's cellular respiration. If the oil tube turns yellow, that's a fermentative reaction. If the tubes are green, the organism couldn't use it, couldn't do anything with the glucose. The conditions were not right. If the media turns blue, again, it couldn't use the glucose, so it used the peptone instead. All right. So let's take a look at our organisms. We're going to use 3, and we're going to run a pair of tubes for each one. First is our old friend, E. coli. Remember, E. coli is a gram-negative rod. That's its gram status. And if you remember from our oxygen lab, E. coli is a facultative anaerobe. Remember, this is the oxygen category that most bacteria belong to. And that means, if you can dig back this far in your memory, a facultative anaerobe prefers to be in oxygen. It will grow best in the presence of oxygen, but it can also grow in an anaerobic condition with no oxygen, and it can grow with low levels of oxygen. So in a thioglycolate tube, you're going to see most growth at the top in the oxic zone, but there will also be growth all the way down to the bottom of the tube in those strings. That's how a facultative anaerobe grows inside of a thioglycolate tube. And E. coli is a facultative anaerobe. So let's skip this question for just a minute and look at the other 2 organisms. We're also going to use Bacillus subtilis. You've actually seen this organism back at the beginning of the course when we were talking about gram staining. Bacillus subtilis is a gram-positive rod. And our third organism is Pseudomonas aeruginosa. We actually talked about this organism when we were talking about microbes in the environment. This is also a gram-negative rod. So 3 organisms to OF test. We're also going to use starch hydrolysis testing. So we will also need some starch agar today. So here's our 2 media that we'll be using. The starch hydrolysis test uses starch agar. And all we're going to do is make a simple streak onto that agar plate. Just one, streak of cells. We're going to incubate, or I should say a zigzag streak of cells. We're going to incubate the starch plates for 24 hours. Then we're going to take them out. We're going to apply the iodine. Don't forget to apply iodine. And then we're going to look for any evidence that the starch was hydrolyzed. For the OF test, remember we're going to use pairs of tubes with OF medium in them. And our OF is going to have glucose as the carbohydrate. We're going to use the semi-solid variety. So we've added just a little bit of agar to the broth to thicken it up a little bit. One tube is going to be inoculated and left open to the air in the incubator, and one is going to be layered over with mineral oil. We're going to leave them to incubate for 24 hours and then look for color changes. Now, here's a good question. We have to have that one OF tube. We have to have one of those tubes open to the air. But we've talked in here about sterile technique, aseptic technique. We can't possibly just put that tube in the incubator open to the air because it's going to get contaminated, right? So what could we do? What could we do with that test tube in order to allow plenty of oxygen in while still keeping contamination from falling in? Any idea how we do that? Remember, these test tubes are typically glass. There are plastic ones as well, but most of them are glass. And they tend to have screw top caps on them. Any idea how you can make a tube open to the air while it's covered? You have any sense of that? Believe it or not, you just place the cap on the tube and you don't screw it down. That's all you have to do. If you take one of those caps and you put it on top of the tube and don't screw it down, that's going to allow plenty of oxygen in there. That's how we get away with it. It's open to the air, but it's got something sitting on the top so we don't get any contamination falling in the tube. There are even caps that are made for test tubes that instead of being screw tops, they're tops that you can press down on the tube, but they have these little ridges on the sides. So even when the top is pressed onto the tube, it's where it belongs on the tube, oxygen can still get in. So we have developed test tubes in the laboratory that we can cover with a cap and still let oxygen get in. Because that's really the only reason to have that tube open to the air, is to make sure oxygen can get in and out. And that way we don't get contamination the next day. All right. I told you a minute ago that OF medium is a basal medium and that we're going to use the semi-solid version. I told you that we take the basal medium and we add glucose to it, lots and lots of glucose, and we add a little bit of peptone to it. And if we're doing the semi-solid version, we're going to add a little bit of agar as well. So this is our ingredient list. Here's that peptone. Remember, another word for peptone that you'll see is tryptone. Those are the same thing. Here's some sodium chloride that we put in, not very much, but organisms have a requirement for salt like we do. Here's our glucose, lots of glucose, 10 grams of glucose. Here's our indicator dye, bromethymol blue. Here's that little bit of agar we're going to put in to thicken up the medium. And here's dipotassium phosphate. This is some mineral that we add to this medium. So take a look at this list and think for a minute why we can only test for aerobic cellular respiration. The difference between aerobic and anaerobic cellular respiration is the final electron acceptor molecule. Aerobes use oxygen as their final electron acceptor. Anaerobes use something like nitrate or sulfate or carbonate, and there are some others. We don't have to add oxygen into the medium, right? Because oxygen's in the air. Oxygen's going to get into the medium from the air. But if you look at this recipe, there's no nitrate in it. There's no carbonate. There's no sulfate. That's why we can't test for anaerobic cellular respiration. There's no final electron acceptor available. The only way that an anaerobe, somebody who did anaerobic cellular respiration, the only way they could grow in that tube is if we gave them their final electron acceptor, and there's none of them in there. We can look at these recipes sometimes, and we can predict what can and can't grow in there. Take a look at that recipe again. Peptone, sodium chloride, glucose, bromethymol blue, agar, dipotassium phosphate. Do you think a fastidious microbe could grow in here? Could a fastidious microbe grow in this? What do you know about those? What do they typically require in a culture medium? Put it in the chat if you think you know. We talked about how some media can grow non-fastidious bacteria, and others can grow fastidious bacteria. What do we add to culture media if we're trying to grow fastidious bacteria? Anybody remember? It's a kind of culture media that it gets its own category. Anybody remember? Fastidious, remember, means they have some weird, unusual nutritional requirement. They're not easy to grow. Can we typically grow fastidious organisms in basal media? Or do we need a different kind? Anybody remember? You're right on the right track, Debbie. They don't necessarily need blood, but they need a complex ingredient. So remember, the kind of media that we use for fastidious organisms is complex media. And the other term we use is chemically undefined, because we're going to add something like blood, or we might add milk, or we might add egg, or meat, or fish product even. We're going to add some ingredient in there that's very complex nutritionally. So A fastidious microbe won't grow in OF medium because it doesn't have any kind of complex ingredients in it. It's a basal medium. It's chemically defined. So we have to remember that. We have to remember that if we try to test for carbohydrate catabolism and we've got a fastidious organism, we can't even grow it, let alone test it. You see what I'm saying? You have to remember that. The OF test has this limitation to it. Fastidious bacteria aren't going to grow in this stuff, period, let alone break down the glucose. Okay, just an important thing to remember. All right, let's look at some results. Here's our starch hydrolysis test. So we pulled the plates out of the incubator the next day and we poured some iodine on them. Here's E. coli. You can just see the streak lines for the E. coli. You can just see the reflection of the light on the streak lines. It is growing, but boy, that plate is black. This plate is also black, even though we can see the microbe growing much better. This is the Pseudomonas plate. And here is Bacillus subtilis. That's a nice test right there. So, which organism is capable of secreting amylase and breaking down starch? Which one of these three? Remember, the iodine binds to starch and turns it black. So anywhere the agar is black, there is starch present.
So the B subtilis.
That's right. This organism, the Bacillus subtilis organism, has done something to the starch. It has broken it down. So if I were to take a little bit of this clear area and take it into the lab and look at the agar itself, there would be no starch in it. There would be lots of glucose in it though, because the amylase breaks starch down into glucose. So this organism that's growing on the streak line is secreting amylase, and the amylase is breaking the starch down into glucose. Iodine doesn't turn glucose black, it turns starch black. So that's a positive test right there. These two are negative. That's a pretty easy test to read and interpret. This one's a little bit harder though. All right, let's take a look. Let's take a look at what we got. So we have 3 pairs of tubes because you always run OF in pairs. Remember, this was the chart we looked at earlier to see an organism that couldn't use the glucose, or an organism that can do oxidative but not fermentative, or an organism that can do both oxidative and fermentative. So remember, when you pull the OF tubes out of the incubator the next day, after they've had a chance to grow, it's the color. It's the color that you're interested in. Now, if you look at, oh, I'm sorry, my labels have moved over because I enlarged the picture. Let's see if I can fit these. These 2 tubes were inoculated with E. coli. These 2 tubes were inoculated with Pseudomonas, and these 2 with Bacillus subtilis. So let's do the E. coli first. 2 yellow tubes. What do I now know about my E. coli? I know that it can do oxidative catabolism of glucose. And to you and I, that means aerobic cellular respiration. So it can do cellular respiration using oxygen as a final electron acceptor. It took that glucose in that open air tube and it broke it down step by step by step until all that was left was carbon dioxide. And then it took all the electrons that it got out of those broken covalent bonds and it uses them to build ATP. Meanwhile, all of that carbon dioxide combined with water and became carbonic acid and turned that open air tube yellow. The fermentative tube is also yellow though, right? The fermentative tube is also yellow, so that E. coli can also ferment glucose. We don't know exactly what it turned the glucose into. We don't know what fermentation pathway it used, but we know it can ferment glucose. And we know that one of the products of that fermentation is acid because the tube turned yellow. This makes sense if you think about what we know about E. coli. E. coli is a facultative anaerobe. E. coli grows best in the presence of oxygen. It prefers to do aerobic cellular respiration, but it doesn't have to. If there's no oxygen around, it just switches pathways. It does another pathway. If it needs to ferment, it ferments. Very, very adaptive organism, a facultative anaerobe, like E. coli. Now, what about the Pseudomonas? Actually, let's look at the bacillus first. Here's that blue color that we talked about. It doesn't go all the way through the tube, but it is there. We don't see yellow in either tube. So Bacillus subtilis cannot do what E. coli can do. It cannot break down glucose using oxidative pathways, and it cannot break down glucose using fermentative pathways under these conditions anyway. But we know it can make use of the peptone. It will typically only make use of the peptone if it's struggling to survive. So that's an important thing to know about the bacillus. When you put bacillus into an open air tube with glucose, it can't do anything with the glucose to make ATP. In fact, it's at risk of dying. So it uses the peptone just to stay alive, just to temporarily stay alive. But it is an organism that cannot do oxidative catabolism with oxygen as the electron acceptor. So it's likely that Bacillus subtilis needs to do anaerobic cellular respiration, and it can't because there's no final electron acceptor for it in that medium. It also can't ferment glucose, which is interesting. Now, what about the Pseudomonas? Because that's the strangest tube. The Pseudomonas clearly cannot ferment glucose. The media is still green. Okay, but look at the oxidative tube. The oxidative tube is partially yellow. It's not yellow all the way down, but it's yellow at the top. Remember, this top part of the tube is going to have the most oxygen in it. So what the heck could be going on in this tube? Well, anytime you pull an OF tube out of the incubator and it's partially yellow, all you need to do is put it back in the incubator for a little while. What this means is that it has started to break down the glucose using oxidative pathways, but it hasn't been doing it for very long. In other words, there's not a lot of acid in here yet. If you put it back in the incubator and take it out a little while later, you're going to see something more like this. The whole tube will be yellow later on. It's just a feature of Pseudomonas that it tends to grow slowly. Here's a nice example of Pseudomonas over time. So these are 3 sets of tubes, all inoculated with Pseudomonas. In this set, it was incubated for 24 hours. And as you can see, part of this open air tube is yellow and part is green. If you put it in for 48 hours, it'll typically turn about half of the tube yellow. It can take up to 5 days in the incubator for that entire tube to turn yellow. Pseudomonas grows really slowly in this medium. There are other media that will encourage Pseudomonas to grow faster. But in OF, which remember is just a basal media with a bunch of glucose in it, Pseudomonas just can't get all of its nutritional needs met very quickly, so it grows very slowly. And if we see that half yellow or quarter yellow, that's a signal to us to just put it back in the incubator. Put it back in, let it incubate a little more, and see if the whole tube turns yellow, because it should. It should if it's using oxidative catabolism of glucose. So when you look at a pair of OF tubes, you evaluate the color. You first look for the color of the tube and which tube. So the yellow color means acidic, and that tells us that the carbohydrate, the glucose, has been catabolized. It has been broken down. That's what the yellow color means. A blue color tells us that the conditions are alkaline, that the glucose was not broken down, and that this organism, in fact, had to use the peptone. It had to metabolize the peptone in order to survive. If the tubes remain green after 24 hours of incubation, we don't see any metabolism of carbohydrate or peptone. And that organism is almost certainly fastidious. If it can't do either pathway, it may be that the reason is because it's fastidious. There's something it needs in order to grow that's not in the media. So it's not going to do either pathway. So color first, evaluate the color first, then compare the open versus closed. Because the open tube is our evaluation of oxidative catabolism, of aerobic cellular respiration. The oil-covered tube is our evaluation of fermentative catabolism. The color changes are the same. If fermentative catabolism occurs in that oil-covered tube, it means that the glucose was fermented and produced an acid byproduct, turning the media yellow. So E. coli was able to do oxidative catabolism of glucose and fermentative catabolism of glucose. Pseudomonas did oxidative catabolism of glucose slowly, but did not do fermentative. Bacillus could not do oxidative catabolism of glucose, but was able to use the peptone to stay alive. So that tells us that this is probably not a fastidious organism, this bacillus. It was not able to do fermentative catabolism of glucose. We would expect that if we put this tube back in the incubator for a couple more days, maybe, the whole tube would turn yellow. So I've got a list of the questions that you should be able to answer now, looking at those results. Which organism or organisms can secrete amylase to digest starch? Which ones can aerobically oxidize or do aerobic cellular respiration of glucose? Which ones can ferment glucose and produce an acid, and which ones are unable to catabolize glucose under these conditions. You should be able to answer those now. Bacillus subtilis is an interesting organism because it has this ability to break down starch. It turns out that a lot of bacteria don't have this. It's just not something they do. They don't secrete amylase. So when you look at textbook reference tables, one of the things that we can find is the starch hydrolysis test and the OF test. And you can look across these tables at a whole lot of different organisms, and you can find out which ones can do which test. What we're going to be doing in a couple of weeks for sort of a final lab assignment is I'm going to give you a table of test results. And I'm going to send you off to look at some reference tables and try to identify a microbe. So you're going to see things in there like whether it has a positive catalase test and whether or not it grows on mannitol salt agar, whether it can grow on EMB and what color it grows and what color it turns the media. And we'll have results for the starch hydrolysis test, and we'll have results for the OF test. So I'm going to give you a whole bunch of data. And then I'm going to ask you to look at some of these reference tables and try to figure out what it is, what organism you have. I'll even give you a clinical scenario. I'll tell you where the sample came from and what signs and symptoms the person had. So you can also use that information as part of your data set. That'll be in a couple weeks after we've talked through a couple more tests. All right, any questions? We've talked about a lot of vocabulary today, and I know it might be new to you, a lot of terminology today. But hopefully, as you work through your lecture material this week, some of this will start to fall into place for you. You'll get a little more comfortable with what's happening in aerobic cellular respiration and anaerobic cellular respiration and fermentation. Hopefully you'll get a little bit more comfortable with those pathways. We don't learn every little detail of those metabolic pathways in this course, but you really don't need to if you understand what the pathways are all about. Cellular respiration makes ATP, and it makes it very efficiently. Fermentation also makes ATP, it just doesn't make it efficiently. What fermentation does make efficiently is alcohol, acid, and or gas. That's what fermentation makes. It just happens to make a little ATP along the way. But respiration is all about making ATP because ATP is something that all living cells have to have or they'll die. And unfortunately, cells do not store large amounts of it. So they have to continuously make it. All right, questions? All right, ladies, I'm going to let you go. I will see you again on Wednesday. Let me know if you have any questions that come up before then, but I will see you on Wednesday.
Okay, thank you.
You're welcome. Have a good evening.
You too.